Now it's my pleasure to introduce to you Professor Dimitri Atiakshin. He is a wonderful person. We were able to collaborate on a paper together and had a wonderful visit while in China. I will read his bio now. He is an advisor to the rector of RUDN University, director of the Scientific and Educational Resource Center, Innovative Technologies of the Immunophenotyping, Digital Spatial Profiling, and Ultrastructural Analysis of Molecular Morphology, RUDN University, Doctor of Medical Sciences, Associate Professor. His professional skills include molecular histology, histochemistry, monoplex and multiple immunochemistry, spatial phenotyping of the tissue microenvironment, spatial proteomics and interactomics, cytology, transmission immunoelectron microscopy, bioinformatics analysis. His area of scientific interest are functional morphology of the tissue microenvironment during adaptation and pathology, inflammation, oncogenesis, fibrosis, spatial profiling of the immune and stromal landscape, biology of mast cells, remodeling of connective tissues, fibrillogenesis of collagen, intracellular signaling and space biomedicine, hydrogen biomedicine, regenerative biomedicine. Now, please welcome Professor Dimitri.
Dear colleagues, I'm very pleased to present new data about some new biological effects of molecular hydrogen. I thank the Summit Organizing Committee for the opportunity to speak and share the results of study devoted to analysis of molecular hydrogen remodeling mechanism on connective tissue and extracellular matrix. During my report, I'm going to talk about anti-fibrotic influence of molecular hydrogen, which was studied in a rodent model. This study was mainly carried out at the People's Friendship University of Russia. Experiments on modeling the inflammation in the lungs and the modifying effects of molecular hydrogen were organized with the help of Professor Oleg Medvedev. The molecular, morphological, and bioinformatic analysis of the biomaterial of lungs was made at the Scientific and Research Center Innovative Technology of Immunophenotyping, Digital Spatial Profiling, and Ultrastructural Analysis of the People's Friendship University of Russia. The small size of molecular hydrogen, its low mass, neutral charge, combined with a high diffusion rate, allow it to easily penetrate cell biomembranes and exerts many biological effects. It has been shown that molecular hydrogen can reduce inflammation, regulate immunocompetent T cells, modulate cell death pathways as well as other signaling pathways, and has mitochondrial effects. Until now, we have a question: how molecular hydrogen can initiate these biological effects remains without answer. The idea of implementing the molecular hydrogen biological effects on the structures of a specific tissue microenvironment through its impact on mast cells seems pathogenetically validated. We know that mast cells may be one of the important targets of molecular hydrogen. In the local tissue microenvironment, including lungs, mast cells play a key role in regulating the local tissue microenvironment, and mast cells have a wide range of receptors allowing to provide high sensitivity for various stimuli of tissue microenvironment. Therefore, mast cells may be a specific target for molecular hydrogen and for formation of its biological effects, including anti-inflammatory, anti-apoptotic, neuroprotective, radioprotective, adaptive, homeostatic, and finally remodeling effects. Today, there are several studies in relation to molecular hydrogen effects on the state of mast cells. However, there is evidently insufficient to reveal the real potential of mast cells in the mediating of biological effects molecular hydrogen. So the aim of the study was to investigate the role of mast cells in development biological effects of molecular hydrogen inhaled in the lungs of rats under simulated pulmonary hypertension, in particularly the reduction of the fibrotic phenotype of the local tissue microenvironment. The experiment included a molecular and histological study of the lung stroma, mast cells, collagen and elastic fibers, and many other. 3 groups were formed: a control group, a group modeling lung inflammation using a monocrotaline model, and a group with using molecular hydrogen as a modifier of the effects of monocrotaline. Molecular hydrogen was used is inhalated form as shown in this slide. To obtain the results, we used histochemical staining, monoplex and multiplex immunohistochemical staining, as well as combined staining methods when traditional histochemical methods were combined with immunohistochemical detection technology of molecular targets. In doing so, we were the first to develop several staining protocols that allows us to simultaneously see several molecular targets and tissue structures on the same histological section of lung. For example, mast cell metachromasia was combined with immunohistochemical detection of tryptase. In addition, histochemical detection of collagen and elastical fibers was combined with immunohistochemical staining of tryptase. These methods allows us to obtain unique data on the remodeling effects of mast cells for the first time. Quantitative analysis of the studied criteria involved a total section of the lungs in the same plane. Planimetric analysis was performed to identify the number of mast cells per unit area of the lungs, the absolute and relative number of mast cells and other cells, the ratio of the elastic and collagen fibers areas, and to specify the profile of mast cell tryptase with determination of high, moderate, and low expression levels and metachromasia using the QPath software product. So, our results. First of all, we found that the monocrotaline led to significant increase in number of lung mast cells in compared to the control. The use of molecular hydrogen suppressed this effect. You see a small number of mast cells and their interaction with the microcirculation bed with a feature of secretion, active secretion. This slide shows the interaction of mast cells with pulmonary fibroblast, and active secretion of granules is clear visible. Exposure to monocrotaline caused an increase in the intrapulmonary mast cells population. The formation of mast cell groups can also be seen, as well as increase in secretion and contact between mast cells and leukocytes is detected, enhancing the inflammatory response of mast cell. The frequency of contact with plasma cells increased. You can see here, and you can see the feature of active secretion. In this slide, we see signs of most likely mitosis as evidenced by the exit of nuclear structures from the cytoplasm to the extracellular matrix. Hydrogen inhalation resulted in decrease in the number of mast cells in the lungs, but loci with increased mast cell content remained. In the lungs, the number of leukocytes and the frequency of contact of mast cells with other immunocompetent cells decreased. Study of tryptase profile in mast cells showed the following results. Monocrotaline resulted in a significant increase in tryptase-positive mast cells, whereas hydrogen inhalation significantly reduced their number. In contrast, the number of metachromatic mast cells without tryptase increased compared with monocrotaline in exposure. Mast cell tryptase level were reduced compared to control in both the monocrotaline and the molecular hydrogen group. In the control group, a certain pool of mast cells without tryptase can be seen. You can see violet just violet cytoplasm of this cell. And here, apparently, this type of mast cell has its special biological characteristics and may represent a reserve for enhancing the expression of specific proteases in all mast cell population. The colocalization of mast cells with different levels of tryptase is clearly visible on this photo. You can see mast cell with high, with medium, modern, and complete absence of this protease. You can see the exocytosis of secretory granules, both with tryptase, here and here and here with tryptase, brown color, and without tryptase. Note that after monocrotaline administration, almost all mast cells began to express tryptase. All mast cells are brown color. At the same time, tryptase could be detected in the nuclei of neighboring cells, exerting epigenetic effects. The effects of tryptase on histones have been shown before. It's possible that monocrotaline resulted in an increase in tryptase turnover between contacting mast cells. You can see here, and it led to enhance pro-inflammatory effects. And inhalation of molecular hydrogen resulted in decreased tryptase expression in mast cells compared to monocrotaline exposure. And also increased secretion of granules without tryptase. You can see here on next slide, you can see secretion of granule without tryptase, violet color. Confocal microscopy allows us to see different levels of tryptase in mast cell granules, from high to low, and large tryptase positive granules are located predominantly in a perinuclear region. The peripheral localization of tryptase in secretory granules you can see very clear. When exposed to monocrotaline, the frequency of intercellular interaction between mast cells increase. And possibly this apparently enhances the pro-inflammatory potential of mast cells. Secretory granules decrease in size. Peripheral intragranular location of tryptase is maintained. Contact of mast cells with each other, it's the feature of interaction of mast cells with each other. Extracellular structures produced by mast cells, you can see here blue color, similar to the extracellular traps described for neutrophils. This phenomenon is called netosis in mast cells. Exposure to molecular hydrogen doesn't alter intracellular distribution of tryptase in mast cells compared to monocrotaline and control groups. Rare secretory granules with high tryptase contact distant from the nucleus. Measurement of the total amount of collagen fibers in whole lung sections showed that monocrotaline leads to a significant increase in the fibrous component of the extracellular matrix of the lungs, whereas inhalation of molecular hydrogen reduced this indicator. An important indicator is the content of reticular fibers, which are known to be immature collagen fibers. As you can see, monocrotaline led to a sharp increase in the number of reticular fibers in the lungs, while exposure to molecular hydrogen decreased this value, reaching to the control level. Collagen fiber profiling showed that molecular hydrogen inhalation prevents the formation of immature collagen fibers in the lung stroma, which may indicate an anti-fibrotic effects of molecular. Reticular fibers are practically not defined in the control group. They are visible in small quantities, and after administration of monocrotaline, a large number of impregnated fibers are detected. They are black color. After using molecular hydrogen, the number of impregnated fibers is reduced compared to the monocrotaline group. Also, individual loci of increased reticular fiber content remain. A similar dynamic compared to the content of collagen fibers is observed with elastic fibers. You can see clearly that monocrotaline leads to increase in the formation of elastic fibers, while molecular hydrogen prevents this. You can see colocalization of elastic and collagen fibers in control group. As a rule, collagen fibers, red color, are larger than elastic fibers, violet color, and have a more linear course. Monocrotaline often caused the formation of in lung areas with a high content of elastic and collagen fibers. The use of molecular hydrogen reduced the number of intrapulmonary loci with increased content of collagen and elastic fibers. We have determined that under the influence of monocrotaline, the frequency of contact of mast cells with reticular fibers increased reliably. Inhalation of molecular hydrogen leads to a decrease in the participation of mast cells in collagen fibrillogenesis, which doesn't reach to a level in the control group. In the control group, mast cells are colocalized predominantly with a mature collagen fibers, which have a yellowish-brown color. You can see here, here, here. And on this slide, you can see a similar picture. The predominant topography in the lungs, where mast cells are colocalized with reticular fibers, is the respiratory section of the lungs, represented by acini. You can see here the place where mast cell can interact with reticular fibers. Monocrotaline leads to a strong increase in the frequency of colocalization, mast cells, and impregnated fibers. You can see black fibers, black color in this photograph picture and in this photo. Similar situation, mast cells are very often in contact with impregnated fibers. You can see a high number of reticular fibers in contact with mast cells, very closely contact. The use of molecular hydrogen resulted in a decrease in the frequency of mast cell contact with reticular fibers. Of course, foci of increased collagen fibrillogenesis remain, and we can see it, we find it very often, but not often how we can see it in a group with use monocrotaline. On this slide, you can see that molecular hydrogen didn't significantly modify the frequency of mast cell adhering to elastic fibers, which was significantly higher than the control values. In controls, colocalization of mast cells with elastic fibers is often observed in the composition of the blood vessel wall. Monocrotaline resulted in the formation of elastic fibers in foci of severe inflammation, where mast cells were colocalized with it. The elastic fibers were of different caliber, both thin and thick. You can see the adhesion of mast cells to both the thin and thick elastic fibers. Sometimes mast cells came into contact with a network of elastic fibers. In this case, targeted secretion of tryptase directly to elastic fibers is clearly visible. Active secretion of tryptase is also observed when using molecular hydrogen. You can see here and here, and you can see the interaction of mast cell with several elastic fibers, which indicates the participation of mast cells in the tensometry of the tissue microenvironment. And here you can see active secretion of tryptase to elastic fiber with tryptase spreading along the fiber, elastic fiber. The next indicator that we consider very important is the level of expression of transforming growth factor beta, TGF beta, in mast cells. You can see that average across the entire mast cell population, this is reduced by molecular hydrogen compared to monocrotaline alone. However, the level of control is not achieved. This is clearly seen when profiling the mast cell population for TGF beta levels. Molecular hydrogen caused a decrease in the level of this growth factor in mast cells compared to monocrotaline group. In this slide, you can see that in the tissue microenvironment of the lungs, a large proportion of mast cells contain TGF beta. And similar condition here, you can see TGF beta positive mast cells in the lung in high number. This micrograph clearly shows that after exposure to monocrotaline, the number of cells positive to TGF beta, green color, increased in the lungs, and the frequency of mast cell contact with the cells also increased. Similar situation of the expression of TGF beta in lung cells you can see on this slide. Exposure to molecular hydrogen reduced the number of mast cells with high expression of transforming growth factor beta. So, our study has shown that mast cells discovered many years ago by Nobel Prize winner Paul Ehrlich can change their biological properties when exposed to molecular hydrogen. This causes a change in their secretory activity and a change in target effects on some structures in the tissue microenvironment and some cells. As you can see in this slide, mast cells can affect to entire cellular landscape. Immune cells, non-immune cells, stromal cells, extracellular matrix, and so on. Molecular hydrogen can arrest the secretion of various mast cell mediators that have pro-inflammatory activity, for example, tryptase, chymase, carboxypeptidase A3, and so on. As for fibrillogenesis, we can highlight the direct effects of mast cells on the activation of the assembly of collagen microfibrils and fibrils. And there are also indirect effects on the enhancement of collagen production through the activity of other cells, for example, fibroblast, myofibroblast, and many other cells. This is ultrastructural evidence to indirect effects of mast cells on fibrillogenesis. In this slide, you can see evidence of direct involvement of mast cells in the formation of collagen fibrils. You can see the target secretion from the mast cell granule to the collagen microfibrils in the area of molecular assembly of collagen fibers. This electronogram was obtained using transmission electron microscopy. So, some conclusions. This study demonstrated the anti-fibrotic effects of molecular hydrogen on the local tissue microenvironment on the lungs, first. Second, this was mediated by the secretory pathways of mast cells in relation to the structural components of the extracellular matrix and other cellular subpopulations. Detected changes in the mast cells phenotype under simulated monocrotaline pulmonary hypertension with inhaled molecular hydrogen delivery allow us to conclude that the molecular phenotype, migratory activity, and the secretory activity of mast cells are sufficiently sensitive to the effects of molecular hydrogen. Decrease in the intra-organ population of tryptase-positive mast cells in the lungs, combined with a decreased efficiency of collagen fibrillogenesis, are important factors that contribute to the preventive effects of molecular hydrogen against inflammatory and profibrogenic states of the lungs. And finally, mast cells as a target of molecular hydrogen is a promising study of further research to identify the new mechanisms is developing biological effects of molecular hydrogen on the living matter. Thank you very much for your attention. See you.